Yeah.
Is that where like the storage is going on? So then would there be more like porphyrin like buildup in?
A different case, remember. This is the
Okay.
pyruvate kinase deficiency, not the porphyria case. So I can go back to this one. So the splenectomy, the idea was, you know, she had symptoms based on lack of red blood cells, right? So she doesn't have enough oxygen delivery to her tissue. And you can correct that through transfusions, but there are complications with transfusions, and you don't want a young 2-year-old kid having to go to the hospital every few months to get one, right? So the basic idea is you want to increase the number of red blood cells that are there. You're not correcting the enzymopathy, but you're just increasing the number of red blood cells that are there. Because the reason that these red blood cells are decreasing in this anemic episode is because they're being taken up in the reticuloendothelial system in the spleen and being eaten up by macrophages and chewed up. Okay, so without the spleen there, red blood cells will still turn over and still be taken up by macrophages, but it will be more distributed throughout the body, and it's going to be a lot slower. So the rationale here is that the rate of red blood cell production is the same, but we're extending the life of those kind of, marginal red blood cells that are there. Okay, does that seem reasonable?
Will they still need like a transfusion instead of 2 months, every year? Or?
Yeah, that's, I'm not sure. They did the splenectomy and then they wrote the case reports, and I didn't see any follow-up on it since then. But yeah, I think that it would be a patient that you would want to regularly check hematocrit on. Okay, good question. All right, so where were we? We were talking about our different disorders of heme synthesis, and we were going south of aminolevulinic acid synthase because we had already talked about that one, right? Okay, and then, I don't know, I've probably said this like 6 times now, but the key feature that drives these porphyrias is the absence of the negative feedback regulation by heme. So what common strategy for treatment of porphyrias is to use endogenous heme to try to restore that negative inhibition of ALAS, or to do just bloodletting, or remove the precursors of or the intermediates in the pathway through phlebotomy. And then there's a kind of a more interesting technique that we'll discuss in a few moments here. Okay, so let's see. We talked about aminolevulinic acid synthase and the disorders there. Let's move down to the next enzyme in the pathway, the aminolevulinic acid dehydratase. And there is an inborn error of metabolism called DOS porphyria, or dehydratase porphyria, that results in loss of activity of this enzyme. In this porphyria, the symptoms are caused by the buildup of the delta aminolevulinic acid. So in this case, would we get blistering of skin in sun-exposed areas? Unlikely, because this is way upstream from the generation of those porphyrinogens. Okay. So yeah, this is neuropathy and abdominal pain are the 2 hallmark symptoms of the dehydratase porphyria. Now, this dehydratase porphyria is quite rare relative to some of the other porphyrias. However, lead exposure is common, and one of the targets of lead is this dehydratase enzyme. And lead poisoning recapitulates some of the symptoms of the DOS porphyria, in that the symptoms are largely neurological and gastrointestinal. Okay, and we also can get an anemia from lead poisoning as well, because you're eliminating heme synthesis. So yeah, lead is a common industrial pollutant, less so in our country than it was 50 years ago, but still prevalent, and in other countries around the world, it's much more so. Okay, so if we look at an in vitro assay where we interrogate the activity of the ALA dehydratase in the presence of different concentrations of its substrate, aminolevulinic acid, we get this slope suggesting this relationship between enzyme activity and substrate concentration. And when we express the Michaelis-Menten data like this, we can calculate the maximum velocity of the enzyme, and we can calculate the Michaelis constant, right? Okay, and when we have lead in the system, that causes a change in those kinetic parameters, right? Okay, so it's probably not an activator, it's probably an inhibitor, and it probably has some particular mechanism that might be of interest to you. Now, we do have within our blood a protein called lead binding protein, or the lead BP, and when you do the same in vitro assay looking at the dehydratase activity in the presence of that lead binding protein, the lead binding protein can somewhat ameliorate the effects of the enzyme activity of lead. Okay, so it's like I guess a natural defense against lead toxicity within our body. All right, now if we look at in a little bit closer detail, so this is looking at the activity of the dehydratase. If we go in and look at it at the quantum level, in a person who is not exposed to lead, the dehydratase enzyme is holding its substrate, aminolevulinic acid, shown here in pink. There is a lysine amino acid that is kind of grabbing onto that aminolevulinic acid substrate and holding it, and then there's another aminolevulinic acid substrate that's binding, not shown here. This orientation of the aminolevulinic acid is dependent on a zinc atom that is stabilized by 3 cysteine side chains. Okay? So that's, not super exciting, but that's just the normal catalytic site of the enzyme. Lead displaces that zinc and occupies those cysteine binding sites, and when you get lead there instead of zinc there, that changes the quantum environment in that catalytic site, and the aminolevulinic acid is no longer able to bind. Okay? All right. Okay, so any questions on the dehydratase? So we've got an inborn error of metabolism that's rare, and then lead poisoning that is not rare. Let's move down to the next enzyme, the porphobilinogen deaminase. And inborn errors of the deaminase enzyme result in acute intermittent porphyria, and this is the most common of the porphyrias that's caused by an inborn error of metabolism. So in the AIP, it is caused by this loss of function mutations in the deaminase enzyme. However, there is also an environmental component, and certainly a component of gender, where women are affected much more frequently than men, and these intermittent attacks are more common in women after puberty and before menopause. So there's a strong component for estrogen exposure as being a precipitating factor for the acute AIP attacks. Beyond just estrogen exposure, any drug exposure that can have that results in oxidative stress, viral infections can also kind of precipitate these episodes. Let's see. So in the baseline kind of levels of the aminolevulinic acid and the porphobilinogen in people with the underlying mutations will be slightly elevated, and then during, just as in the absence of the AIP attack, And then during an AIP crisis, those levels will greatly increase in the urine. Okay, so that's going to be one way that you can diagnose it. You can also diagnose it by looking at the enzyme activity or by doing a genetics analysis. Okay, so treatments, well, let's talk about, besides just bloodletting or treatment with intravenous heme, talk about something that's a little more interesting and possibly more durable of a cure. Is this something that you've seen before? Okay, so kind of a trivial detail, but best answer here is the dicer. So a dicer is processing it, and then the risk complex is displaying it. Okay, so dicer, best answer here. And why I'm asking this question kind of seemingly out of the blue is that this is this system of intracellular antiviral defense. This can be exploited for the treatments of AIP and potentially for other conditions as well. And this is a drug called Givirisan, which was approved by the FDA a few years ago and has shown some pretty promising results in AIP patients. So the Givirisan is a RNA that interferes with the expression of ALAS1. Yeah, I get so these porphyrias. Some of them are due to the erythroid compartment, and some of them can be due to symptoms from the hepatic compartment. In the case of the AIP, the main issue is with the hepatic production of the ALA and the porphobilinogen. So the siRNA gets into the cells and it's recognized as an endogenous RNA and is just processed by the RISC complex, or excuse me, processed by dicer, and then it's displayed by the RISC complex, and then any other RNA that the cell is making that is homologous to that RNA that you put into the cell, that will then be degraded. So this is a way to decrease the expression of an endogenous gene, and then that will slow the rate of substrates going into the heme synthesis pathway, because that's the rate limiting step. So as far as how that RNA gets into the cell, the RNA is linked to galactose, and then it's taken up. So there's a galactose receptor on cells that will bind it and recognize it and take it up through receptor-mediated endocytosis. So that part is really similar to the RNA vaccines that are developed against COVID and potentially against a lot of other things, is by linking that nucleic acid to sugar, and then that allows it to enter into the cell. Okay, so just to continue with the story, phase 1 trials of the Givirisan showed some dramatic biochemical improvements if you're looking at the level of mRNA in the urine or the levels of the porphobilinogen or aminolevulinic acid in the urine. This is top line is control, and then there are different doses of the Givirisan, and you see a nice dose-dependent inhibition of the accumulation of these different intermediates. So this was a phase 1 trial, and then a phase 3 trial was comparing this against standard of care, and the standard of care being intravenous Heme administered in the hospital. They showed the same biochemical improvements, and then they also showed fewer hospitalizations and fewer less need for intravenous heme. So this is a pretty promising treatment. Yes?
So is the Givirisan, it's preventing the entire heme synthesis pathway to prevent the buildup of intermediates, which is relieving symptoms?
Yes, and it's specific to the ALAS1 in the liver, so it's presumably not going to cause an anemia as well. Yeah.
That was my question. Yeah, so you wouldn't have to supplement with something else to maintain the heme cycle because it's not shutting down the complete heme cycle.
Yeah, the erythropoietic heme compartment, that should be fine. Yeah, good question. Yes.
So the intermittent porphyria, it's like more closely associated with the porphobilinogen deaminase enzyme, but it's like this this givericerin is targeting like the first step. So would any of the other porphyrias be sort of also dealt with because they're at different like enzymatic levels?
Potentially, yes, but AIP is the most common, and that's kind of where, that's where they're targeting, or that's the population that they're going for first because it's a bigger population, I suppose. But potentially, it has the similar aspect for other defects in the heme synthesis pathway. I'm talking about the deaminase deficiency here, but the idea with that therapy, it's not correcting the deaminase deficiency, but it's blocking the amount of substrates that are coming in or slowing the rate of the substrates coming into the pathway, and then you don't get the buildup of the toxic ones, the aminolevulinic acids. Okay, is that rationale for that treatment make sense? A little bit? Okay. All right, so let's move down. So I'm going to not hit every one of these porphyrias, but I'll just kind of talk about some of the most significant ones. So porphyria cutanea tarda, this is, so acute intermittent porphyria, this is the most common porphyria. Porphyria cutanea tarda is the most common non-acute porphyria, okay? It's classified as a non-acute porphyria, however, it can result in a sporadic fashion or an intermittent fashion related to oxidative stress. So with the porphyria cutanea tarda, there is an inherited form where there are inherited defects in the uroporphobilinogen decarboxylase. You can also get sporadic form where you get a decreased activity of the decarboxylase in the absence of any apparent mutations. Okay. And I'm going to talk just about that sporadic form and that sporadic form and its relationship to oxidative stress in porphyria cutanea tarda. That is implicated in some of the other, like the episodic nature of some of the other porphyrias as well. Okay, so the decarboxylase enzyme is shown here, and in a normal, healthy hepatocyte, you get the generation of aminolevulinic acid from the glycine and succinyl-CoA. That goes down to the uroporphyrinogen, and the decarboxylase turns it into the coproporphyrinogen. And then that goes down the pathway, and ferrochelatase inserts the iron to make heme, and then you get that feedback inhibition. Okay, so in the context of oxidative stress, and the oxidative stress can occur from diverse sources. It could be from normal metabolism of estrogens, or it could be through pathological exposure to toxin, or it could be from viral exposure, such as the hepatitis C virus. There are kind of diverse forms of oxidative stress, and if we don't have robust enough antioxidant defenses within our cells, you're going to get non-enzymatic oxidation of diverse substrates within the cell. So the upstream intermediates of the heme synthesis pathway are one of the targets for this non-enzymatic oxidation. When the uroporphyrinogen, this can be modified by oxidative stress into not a substrate for the decarboxylase, but actually a molecule that competes for the normal substrate. Okay, so when you get ROS from either, hemochromatosis, viral infection, normal metabolism of estrogen, what have you, that can turn this substrate of the decarboxylase into an inhibitor of the decarboxylase. Okay, and that then blocks the whole pathway. You get more of this building up, and then that becomes more available for the reactive oxygen species to make it into an inhibitor, and it kind of exacerbates the problem into this acute crisis. There is a relationship between iron metabolism and porphyria cutanea tarda, and some of the hemochromatosis genes are shown here. We'll talk more about those on, I think, Tuesday of next week. So there's a relationship between iron exposure and iron overload and generating this oxidative stress. Okay, so in terms of the symptom profile for porphyria cutanea tarda, and then I don't think I mentioned alcohol as one of the things that precipitates porphyria cutanea tarda. So, like normally, estrogen metabolism generates oxidative stress, and so women have kind of a predisposition to porphyria cutanea tarda. However, alcohol is also a huge cause of oxidative stress, and men generally abuse alcohol to a much greater extent than women do. And for that reason, porphyria cutanea tarda is actually more common in men, and it's more, the most common cause is chronic alcohol consumption. Okay. All right, so in terms of the symptom profile, you get the porphyrinogen buildup that can get oxidized by sunlight and cause the blistering agents, or cause the blistering of sun-exposed skin. You also have the porphyrinogens in urine, and these can give it a darker color in normal light, and then, I'm not sure if you can see this, but it has a fluorescent property under UV light, so that can be part of the diagnosis for the porphyria cutanea tarda. Then hirsutism, or more hair growth in the face and periorbital region, that's another common condition of PCT. Okay, so one of the treatments for PCT is just to remove blood from the patients. Now the toxic buildups are being generated by loss of activity in the hepatocyte. But if you remove blood from the patient and increase the rate of erythropoiesis, those intermediates are going to get consumed in the bone marrow and relieve some of the symptoms. So just periodic phlebotomy. This is, going back to 1968, described here, but then it's also, a current strategy as well for treatment of PCT. Okay, yes?
You mentioned bloodletting a lot, how much? Because the average person goes and gives blood and that's a pint. For a treatment of one of these conditions, how much blood gets a result?
I don't think it's much more than that. It's not like you're, severely depleting them.
Yeah.
But the idea is you want to nudge up the rate of erythropoiesis. And I don't think if your bone marrow is working, I don't think you have to take a lot of blood out to do that. So I guess the short answer is I don't know. I'm not a clinician. So, any other questions? Yeah.
So some of these intermediates build up that cause the specific symptoms like GI or neurological, they're all though underlying is the accumulation of iron and heme?
No. Accumulation of iron is a common feature because iron is a substrate for that last enzyme, and if you don't have the other substrate around, then the iron doesn't have a fate and can accumulate. As far as heme, no. I mean, heme is not getting produced, so that's.
Or iron, I should say, specifically iron is accumulating in all of these. regardless of the other specific buildups.
Yes, I think that's safe to say, that the iron doesn't have that important fates. So it's gonna accumulate, and particularly in the erythroid cells, because in the erythroid precursor cells, really the iron isn't doing anything else except being in heme. In a hepatocyte, iron goes, it can go into iron sulfur clusters or ferritin or hemosiderin or something. It has other metabolic fates besides going into heme. Okay, should we look at a couple of problems looking at porphyrias? I've got a couple of clicker slides and explanation, and then I think, oh yeah, we do have to talk about heme breakdown, so I gotta save some time for that. All right, let's look at a couple clicker slides and then kind of break them down here. And go ahead and take a look at the pathway as you're solving this rather than relying on the memory. Okay, ready? Okay, so this would be a clinical presentation that is consistent with the porphyria cutanea tarda. So let's take a look at the question down here. So blistering on her hands and neck exacerbated by exposure to sunlight. So we know that there's a porphyrinogen being generated, right? So that would, the dehydratase deficiency or aminolevulinic acid synthase deficiency, or the uroporphyrinogen co-synthase one. I don't think we talked about that one, but that would be out, right? So it's got to be something downstream of where the porphyrinogens are made. Then she has, let's see, porphobilinogen is elevated. So that is, porphobilinogen is elevated, but protoporphyrinogen 9 is normal. So I think, by exclusion, you would get at the decarboxylase porphyria cutanea tarda. And this is about as hard as we could make one of these questions. I guess I could, instead of UGT1A1, I could make another one of the porphyrias, I guess. But, so think about distinguishing a porphyria from kind of other things rather than one porphyria from another. That said, why don't we try? Okay, so in this one, we don't have any skin blistering, right? No indication of that. So it's probably one of the more upstream enzymes, right? Okay, so this would be most consistent with a deaminase, which is C. Okay, great. So almost majority of you got that right. So there wasn't any lead around, but we have these neurological symptoms, and I think it says that ALA is elevated. Right, so we can presume that ALA dehydratase is Yeah, I guess, okay. And then there's no blistering on the sun-exposed skin, so it's gotta be upstream of the generation of porphyrinogen. Okay, so kind of tough questions, but I think they're solvable. I don't think I gave you this slide or the other explanation one, but I'll make that available for download at the end of class. Okay, so should we switch away from the porphyrias? Okay. So let's talk about heme function and what it does. And that's it, right? Takes oxygen from the lungs, delivers it to tissues, takes carbon dioxide from tissues and delivers it to the lungs. Right? And let's just leave it at that. All right, so let's talk about what happens to heme after it is done. So the average lifespan of a red blood cell is, what I think, 120 days, something like that. And so it lives in the bone marrow and it makes all of its hemoglobin and all of its glycolysis enzymes. Then it leaves the bone marrow, destroys any capacity to synthesize new hemoglobin or new enzymes, floats around the blood doing this, and then it gets degraded. And the degradation of the red blood cells, so there is red blood cell destruction by macrophages in the periphery, and there is hemolysis of red blood cells in the periphery normally. However, most of this is occurring in the spleen, in the reticuloendothelial system. So as the red blood cells are kind of, getting a little ragged from doing their metabolic job and being exposed to all this oxidative stress, as they pass through the spleen, macrophages are going to be taking chunks out of them until there's nothing left. And all of the components of the red blood cell, the protein components in hemoglobin and other enzymes, gets digested down to individual amino acids, and that joins the amino acid pool. The heme component has a dedicated metabolic route of degradation. So first there is a heme oxidase enzyme that breaks open the ring of the heme and decyclizes it and then releases iron. So that's shown here, the heme oxidase enzyme, and this is present within macrophages, and then there can also be an extracellular heme oxidase activity. If you get this extracellular hemolysis of red blood cells, there is a way to, for a macrophage, both to take up extracellular heme and to do the heme oxidase enzyme activity extracellularly. So the heme oxidase enzyme, by breaking that bridge, it is opening up the ring structure and releasing the iron. That bridge that it's breaking, that methylene bridge, that actually becomes a carbon monoxide. So if you at some point, if you're at bar trivia and you want to know, you're asked what's the only source of carbon monoxide within our body, this is it. So we make carbon monoxide a little bit through this pathway. Everything else we get through tailpipe exhaust, I guess. Okay, so the product of heme oxidase, or as shown here, heme oxygenase oxidase, it's frequently called that as well, where you remove this carbon and the iron and decyclize the enzyme, is biliverdin, and biliverdin has a green color. The biliverdin reductase enzyme is saturating this carbon at the center of the molecule to make bilirubin, and bilirubin has a yellow color. So if you've ever seen a really bad bruise where you get this kind of a yellowy, greeny color, that is due to not hemoglobin, heme breakdown, but myoglobin and muscle. That muscle cells will break down, release the myoglobin, then the heme and the myoglobin will be because it's kind of stuck there, not circulating, and it just diffuses out, and as it is, it's being converted to first the biliverdin and then the bilirubin. When this happens in the reticuloendothelial system, or in a red blood cell, you know, typically you're in circulation, so these things don't accumulate to make a bruise every time a red blood cell gets destroyed. Okay? That said, the accumulation of bilirubin, this is the cause of the discoloration in the eyes and the skin that's associated with jaundice. Okay, so it's this molecule is building up, and that can be to a few different reasons, as we'll discuss shortly. Okay, so once bilirubin is generated, bilirubin is transported in the blood from the spleen to the liver, and bilirubin, as you may infer from the structure here, this is a very insoluble molecule, so it's carried to the liver in complex with albumin, right? Okay, the universal transporter of everything within the cell. Okay, and then within the liver, the bilirubin is a substrate for glucuronation, or the addition of glucuronic acid. And the addition of glucuronic acid to the molecule, that makes it way more soluble, and it allows it to be excreted in the bile. Okay, so glucuronic acid, if you remember, like glycogen metabolism and glycosylation reactions, there are these nucleotide conjugated sugars, right? And UDP glucose is the first one. The sugar can be modified to change it from a glucose into changing it into a carboxylic acid called glucuronic acid. So this glucuronic acid, this is still conjugated to the nucleotide. Then a glucuronal transferase enzyme will transfer that sugar component off of the nucleotide and onto some other substrate. Okay, so there is a family of these glucuronal transferase enzymes, and they have different substrate specificity depending on their subunit organization, and there's a specific organization of the glucuronal transferase enzymes that use the bilirubin as a substrate, and those take the bilirubin and add the glucuronic acid to these two methyl groups, and that then increases the solubility of the molecule and allows it to be excreted in the bile ducts. Okay. So I'm going to return to that story of the glucuronic acid transferase enzymes shortly, but I just want to talk about jaundice for a moment. So jaundice occurs when bilirubin builds up, and bilirubin can build up from a few different kind of conceptual reasons. One is you could get excess production of bilirubin, such as with rapid hemolysis. So if you had a rapid lysis of a bunch of blood cells all at once, you would get a lot of bilirubin production, and it would exceed the capacity of the glucuronal transferase systems in your liver. Okay, so that would be an example of hemolytic jaundice. You can also get hepatic jaundice that results from just a normal rate of production of bilirubin from a normal rate of red blood cell turnover. But if you have some deficiency, some just liver failure from chronic alcoholism, for example, you're going to have not as much capacity in your liver to do that glucuronal transferase function, and you're going to get a buildup of bilirubin. So that's a case of hepatocellular jaundice, and really anything that compromises the function of the liver could potentially result in jaundice. Then a third form is kind of related to hepatocellular jaundice, but this is not due to a loss of the biochemical activity of the liver, but due to an obstruction of the way that the glucuronate conjugated bilirubin would be leaving the liver, and that's out through the bile duct. So these three in red, these are like pathological forms of jaundice. Neonatal jaundice is, I guess it's kind of pathological, but it's also fairly frequent and frequently resolves on its own, and this is just due to immaturity of that glucuronate transferase system, and also due to hemolysis that can increase after childbirth. So this can either be not treated or it can be treated with that phototherapy to break open the bilirubin. Okay, so we can evaluate these different forms of jaundice by looking at the different forms of bilirubin. So if we measure just the free bilirubin or the unconjugated bilirubin, this is, you know, as a biochemist, I call it the unconjugated bilirubin, but clinicians have to have different vocabulary for things. So they call it the indirect bilirubin. And then the conjugated, this is referred to in clinical parlance as the direct bilirubin. So if you get, you know, just my kind of crude understanding of this, and hopefully the pathologist will give you more information about this. If you have both the unconjugated and the conjugated increasing kind of at the same amount, that's more likely to be a hemolytic cause of the jaundice. Whereas if you have the unconjugated increasing way more than the conjugated, then that's more likely to be a hepatocellular form. Okay, it kind of makes sense if you think about it. Okay, questions? All right, I'm going to spend the last few moments of the course talking about the enzymes that do that glucuronal transferase activity that is necessary for converting the bilirubin into the bilirubin glucuronate conjugate. So back in the 1950s, there was an inborn error of metabolism that was described called Crigler-Najjar syndrome. And Crigler-Najjar syndrome is a form of jaundice that can result in kernicterus or brain damage due to bilirubin buildup and ultimately death. And the cause of the jaundice was a deficiency in the glucuronal transferase enzyme that was specific for using bilirubin as its substrate. Okay, so if we look at the organization or the gene organization of that glucuronal transferase enzyme, we have the enzymatic exons, or the portion of the enzyme that's actually doing something, that's grabbing onto the UDP glucuronate and cleaving that bond and making a bond with another one. So we've got all these common exons at the 3' end, and then you can make different enzymes with alternate promoter transcriptional start sites. So there's multiple different promoters, and all those promoters they transcribe into a new first exon. So if you get, for example, transcription here, you'll get A1, 2, 3, 4, and 5. If you start transcription at this promoter, you get A2 as the first exon, and then 2, 3, 4, 5, etc. So those first exons, those are providing the substrate specificity for the enzyme. Okay, so if you have the A1, that first exon, if you start from that promoter, then you make an enzyme that grabs onto bilirubin and adds a glucuronic acid to it. If you use those other transcriptional start sites, then the enzyme is going to grab onto some other substrate and add a glucuronic acid onto it. Like, I don't know, acetaminophen, I think, is modified by glucuronation. Then what else? I'm kind of blanking on it, but there are many different substrates that are glucuranated as a way to get rid of them, and this is kind of a elegant way to conserve genetic information by having these common enzymes and then the substrate specificity granted by the alternate promoter start sites. Okay, so in patients with that Crigler-Najjar syndrome, that is due to a specific lack of expression of that A1 form of the glucuronic the glucuronal transferase enzyme. And in those patients, you don't have the glucuronal transferase, so the bilirubin then just never becomes the bilirubin monoglucuronate or the bilirubin diglucuronate as shown here. And the bilirubin then builds up in the blood, and it can cause brain damage and then ultimately death. So this is showing some of the transporters that transport the products into the bile ducts, etc. Not so important. Okay, so fortunately, Crigler-Najjar syndrome is treatable with phenobarbital, and phenobarbital will activate a transcription factor called the constitutive androstenedione receptor, and this is going to increase the expression or increase transcription from that promoter that makes the specific bilirubin-specific glucuronal transferase enzyme. Okay, so Crigler-Najjar, it's not a total loss of this enzyme. You can increase its expression with the phenobarbital. Questions? Okay. This is a kind of nice cartoon showing an overview of heme synthesis and heme breakdown. Generally, we wouldn't be doing these at the same time in the same cell, but it's just for convenience. It's showing the all together there. Okay. Time for a quick case here in a minute and a half. Okay, so works in a pencil factory. What do you think? Lead in pencils is not lead, right? It's graphite. Okay, so I'm gonna kind of rush through to the end of this, I guess, and I'll leave it for you to read the intervening ones. So she had, let's see, liver failure, anemia, sinus tach, and GI symptoms and neurological symptoms. Okay, so lead poisoning. Pencils use graphite, not lead. However, lead is a common industrial toxin, and if you work in a factory, lead exposure, should be on the list no matter what type of a factory. In this case, no lead in the blood or urine. Okay, so a porphyria, the patient's urine darkened, and it had high levels of aminolevulinic acid, but we couldn't do any of the other enzyme activities. So sequencing of the porphyrin deaminase, and I'm not sure why they keyed in on that one, but that's one of the most common, the most common cause of porphyria. They found that in one of the exons there was a mutation in the splice acceptor site. Okay, so remember that GUAG that are the invariant nucleotides on either side of an intron that gets spliced out. So in this patient, one of those was mutated, resulting in a non-functional deaminase enzyme. And that was described here. So just in the interest of time, I think I'll just not hold you over for this, but I'll put those slides from the case in with the download at the end of class as usual. And if you have any questions about it, please let me know. Okay, thank you.
Next class, just email me and ask me how I know what is important to take notes on or not during the lecture. And also if there's a microphone in the she said it's okay if I don't respond until July. Until July. Like it's okay if it takes you a long time to respond, if it's June or even July. I just want to know how you know what's important. Girl, I'll let you know when I know. Yeah. There's a microwave. I can say that. Because I think there's 4 in July. Yeah, there's a microwave. There's a vacuum of the rest.
These are good, but not as good as.
nutrient absorption.
You don't really take notes. I highlight things. I know.
Depends on where the block is, like, because you can test the bile duct.
We're going to the library.
You buy it?
Huh? Yeah, I'm a huge fan.
What is it like? Is it like a bunch of reviews? Oh my god.
What is it?
With 2,376 questions.
I'm down. I'll come.
U-World doesn't have videos, I don't think.
U-World is the question bank. Is really good. Like, this one has questions, but not as many as U-World. But these have like, so I've been doing immunology, obviously. So you like watch them, you watch
They have
the
them,
video.
but ours didn't work when we
They're
did ours.
always shorter than our lectures, but like have the same amount of information, just
I
more
think I have
condensed
those
and
illegally
efficient.
downloaded on my computer.
And then I take
I
like,
think I might have illegally downloaded it.
and then I
too.
take like the post video quiz, and then I go into On King and unsuspend that video and do the cards.
Get a bile
And
duct
this is-
obstruction, like that bile is toxic to the liver, you know, so if that's not corrected, it'll cause.
Go in and check. Go into the room that you're doing it in and check.
I think I pirated the videos too at some point in my life.
Maybe he'd just rather have his
I have
own.
sketching on this too, and I've not used sketching.
You did, okay.
The downside is I don't have the tags, but all these little manual labor.
Actually, there wasn't one.
Okay.
Oh, then they connected.
On King set that up.
Oh,
So you just go under like step 1B11 or 12, whichever deck you use, and then
It's gonna depend on the person, you know, sometimes some of the.
on King
video.
used to be King.
I was deciding if I go home.
How the fucker do that?
She's come here and then.
Well, it wasn't
Yeah, yeah, so, thanks, it's been in the back of my head
collaboration, like
like
you
every day.
can, you can suggest
But if that's on, that's good.
parts, like they basically just
It was like 8
became a
people at the
central
start,
data,
and
yeah.
Now it's like a lot. Yeah.
became a central database for like anyone who's on. He's a resident, he's like a first or second year resident.
So it's like within the last 5 years. Damn, kind of came in the right
I
time.
did see some stuff on the poryphyrias, but not listed.
Thanks, son. Make me a good doctor.
They were just listed under the.
All right, now I'm gonna go see the puppy.
She at your place or are you like going to her?
Her place. She's like, oh, can you stay over at my place? I'm like, no, I gotta go to Kirby too. If I was like, I won't tonight because I got the test to do tomorrow.
Yeah.
But I said later on I will. If you're bored, let me know. She's her the puppy. You want to stay manager before that? Plus, you look at Dakota's place.